All right, so I'm a computational biologist by training, and I'm probably one of the people that is farthest away from the HPC community in some sense, but I'm a heavy consumer of HPC. So I'm going to talk about some of the vignettes that we have in biology that are driven by HPC, and in particular, data analytics and machine learning tools that we are developing in order to address some of the emerging challenges in biology. So there's no need for me to say this here again, but the data sets that you're seeing from biology and biomedical side of research are really exploding. So this is a paper that was published in 2015. When they published, they basically were tracking the number of genomes that we have today and how sequencing is actually going forward in terms of generating data. And although you cannot see it very well, although you cannot see it very well, by 2015 or so, the current capacity is that we've already crossed a couple of exabytes, almost nearly reaching zettabyte data capacities that are needed to be supported to make any inference from it. Coupled to that, if you also start tracking the number of papers that talk about big data, you will start seeing that bioinformatics is actually quite at the bottom of this game. On the other hand, imaging data sets are actually tending to top because imaging also generates equal amounts of data, which has this volume, veracity, and everything that Prabhat was talking about, and the type of attributes that you're working with. The projected growth is about zettabytes within the next 10 years or so, which means the necessary tools and techniques that we need to build have to go into dealing with these types of data sets. What is clearly different from the way we look at astronomy data sets, Twitter and YouTube, which is kind of the industrial heavy duty or heavy lifting data sets, is the fact that genomics data sets tend to be much more noisy. And if you look at the amount of computation that you need to run on them, for example, even questions of saying, let's take a given genome and compare it to all the other genomes that we know on this planet, they can take up quite a lot of computing hours, even on a very large computing platform like Titan at Oak Ridge. So given this type of setup that we have with data, the question now becomes, what are the side of tools that we need to develop in order to analyze these data sets? I'm kind of going to stick towards the side of algorithm development and what we have done in order to handle these problems. So biology at 100,000 feet level is essentially multi-scale. So you start out by talking about a whole number of things. It deals with things that go all the way from nano to the macro scale, which involves nano is at the level of proteins, bacteria, everything that you want to think at the lowest possible level in terms of molecular and cellular interactions. Then going to the next scale where you're looking at genomes and genomic interactions, and then moving further down the lane where you're talking about communities, tissue structures, and organizational structures. And finally, going all the way to the fact of saying you have populations, you have now people that you're caring about, you need to worry about how to treat them and so on. And at each scale, you need to make a jump in terms of how you analyze these data sets and what perspective you get from them. And the key delivery point here is that insights that you derive from this data is the one that is going to ultimately result in better changes. So you might have heard of the precision medicine movement. You might have heard of the big data to knowledge movement that NIH has been pursuing actively. And each of these cases, you need to put yourself in that bucket and say, okay, each of these has unique data challenges. It comes with different sorts of data sets that you need to integrate, you need to work with, and how do you make better decisions based on that types of data. So the key thing to remember from this is biological data sets are big, they're messy in the sense they have a lot of noise, they have missing data, you need to worry about them all the time. They're also heterogeneous, which makes it even harder to kind of build these workflows and try to integrate all these different data sets together in order to get better insights. And we need to build data analytics tools that are essentially statistically aware, which means you use the statistics in the data to guide how you decide on what sort of methods are useful for it. So I'm going to present 4 different vignettes of what we are working on. Fortunately, at Oak Ridge, I have the luxury of working on 4 different scales, at 4 different scales, and trying to look at how we can develop solutions for each of these scales. At the nanoscale, I'm going to talk a little bit about what we are doing with the spallation neutron source data sets and how we are trying to integrate that for cancer biology. At the microscale, we're going to talk a little bit about how we are building large-scale phylogenetic tree construction. In some sense, we want to build a network of life. Think of it as a modern version of tree of life kind of a thing. So we want to do that, and how do we develop such approaches? At the mesoscale, I'm going to talk a little bit about our involvement with the Big Neuron Challenge. This is a challenge that is running across the world right now. It includes Berkeley, ourselves, and a couple of other national labs in the country, as well as across the supercomputing centers in Europe and Asia, and how we are setting up a data analysis challenge across the world to run these types of visualization and analysis of neuron morphology data sets. And the last one I'm going to talk is about the Oak Ridge Biosurveillance Toolkit, where we use electronic healthcare reimbursement data as a proxy for electronic medical records to study disease progression and how do we model that. So with that, let me first get into the first thing that I wanted to talk to you about, which is how are we integrating neutron scattering experiments with molecular simulations using data analytic approaches. So in biology, one of the key drivers is that we need a problem to work on, and one of the problems that is key in cancer is understanding how signaling alters pathways, healthy pathways in the cell, and leads to cancer and types of this one. This class of proteins, the intrinsically disordered proteins that we work with, are kind of an anomaly in the protein world, because I think when you're in grade school or when you're in high school, you're always taught that a single protein codes a single function. So if you know the structure of the function, you can determine what the function of the protein is. But these proteins tend to violate that very principle. They are basically proteins that don't have a stable tertiary structure. They are highly flexible. They have very adaptable binding surfaces. For example, this little protein that is shown here, it's about a segment of about 50 residues or so, binds to 4 other proteins in completely different orientations, which is kind of key because this kind of binding leads to different signaling pathways being activated. And in one case, it so happens that when that binding happens, it can lead to cancer. So you want to be able to dissect these types of mechanisms in these cells, and how do we study that is actually a big question. About 35% of the entire eukaryotic proteome, that means all the proteins that are expressed in humans all the way to eukaryotic cells, including yeast, have disorder, some form of disorder, which means that this is a significant class of proteins and that we don't know much about them. And about 65% of these proteins are involved in some form of diseases, including Alzheimer's, Huntington's, cardiovascular, diabetes, and so on and so forth. So these proteins actually play a major role in the cell in terms of regulating function, but we don't know much about them. And the particular case I'm going to talk about is this little domain, which is a 53 residue protein, nuclear coactivated binding domain. And if you look at the existing literature, all that you know are these different structures where it is bound to these different complexes. So for example, you have a complex with P53 TAD, you have another one which is called ACTAR, the other one is called SRC1 and IRF3. And if you look at each one of them, the last 3 on the right have no essential change in their structure, but if you look at the cartoon for the left-hand most one, you can see that the orientation is completely different. So how do we dissect these mechanisms is actually a big question, and there are lots of scientific questions around it. I won't go into the details of these scientific questions, but the key idea is how do we dissect these mechanisms. At Oak Ridge, we are fortunate to have the largest neutron scattering facilities on the planet. So this is actually a scale diagram of what sort of instruments that are out there in that facility, where we can use these different instruments to measure certain aspects of how these proteins function. And these SANS measurements, or small angle neutron scattering measurements as we talk about, they are quite large in the sense the data sets that they spew out is quite large, but they're extremely sparse. So if you look at what information you get from these, if I take this protein and put it into this instrument and get a reading out, all it is going to tell me is overall shape and size of this protein. It doesn't tell me anything about atomistic details. You have to resolve it yourself. So from the SANS experiment, you're only able to determine the average size and shape of the molecule, but you're not able to say anything about how the atoms or individual atoms are interacting with each other. So how about we use computing to enable this type of challenge where we can improve the resolution of these experiments itself? So SANS is a huge facility. It's a multi-billion-dollar facility. And of course, we have computing at Oak Ridge, which is why we went ahead and started doing these long timescale simulations of these proteins. And in this case, it so happens that these proteins are quite small, and so we can actually simulate them pretty much to millisecond. But if you think of molecular dynamics simulations, this is the bread and butter of all HPC work. And what you have there are, you can have anywhere between order of 10,000 atoms to 1,000,000 atoms in the system. But the problem is you have to integrate over 15 orders of timescales, from femtoseconds all the way to seconds. And you have to develop these efficient algorithms for scaling MD simulations, which we have done, and we have implemented them on these systems. And now we can get millisecond timescale trajectories, which is about 12 orders of magnitude what we could get from 2000s or so within 10 days or so, which is actually quite nice. But the problem is each of these simulations can generate over several terabytes of data in any given single day. So if you're running them continuously, you can be dumping data very fast from these simulations. And now you have this big data challenge of saying, how do I manage my simulation data sets? How do I understand what my simulation data sets are saying? There are some key challenges for data analytics here. One is that this is very high dimensional data. So if you think of tracking just XYZ coordinates of all the atoms in your systems, if you have 1,000,000 atoms in your system, then you're pretty much screwed. At every time step, you're getting 1,000,000 cross 3, and that is tracked over several millions of time steps. And so you essentially end up with a massive volume of data, which you will have to digest and see what you can do. So that high dimensional data is actually quite large. You also have to find these simulations. I mean, you have to analyze these simulations in a way you can get biological insights from them, which can be actually a very small subset of this data, which can be rare events, which can be events that we find interesting and the biologist finds interesting. And then you have to put them together with the experiments. And one of the problems, though, is that you cannot replicate all of these experimental conditions within your solution so that you can do the MD simulation also exactly the same way. So in order to attack some of these problems, we have developed event detection techniques on these massive dimensional data sets, and we are using higher order statistical signatures that automatically extract these signatures in such a way that you can organize these high dimensional data sets. And finally, we are integrating them with Bayesian methods so that you can improve the resolution of these neutron scattering techniques. So let me talk a little bit about what we are doing with these higher order signatures. So if you stand at a particular atom and track how much you're moving with respect to a mean position, this is a depiction of the distributions that you obtain from that. And what you'll observe is that you can see that it is quite long-tailed. And by long-tailed, we mean there's our heaviness on the distributions on either side of a Gaussian tail, which is shown in gray there. And so these long-tail fluctuations are not only common in these MD simulations, they're very common in imaging data sets, astronomy data sets, and so on and so forth. So there's a lot of work being done there in order to extract these signatures. The good thing, though, is that if you take some of these techniques and apply on these systems, now you have a massive analysis problem, which you end up doing anyways, and then project all of these confirmations that you get from the simulation. So what you see there as a cloud is essentially a collection of about 100,000 points that were equally sampled from there. If I were to draw a million by million matrix, then it wouldn't make sense here. So we have sampled about 100,000 points and just projecting them to the lowest dimensional spaces by following these long-tail fluctuations. Once we do that, and you can paint them with internal energy values that you're obtaining from the simulations, or you can use that with other experimental observables, for example, the radius of gyration, And in each case, what I want you to see is that they actually cluster in different forms, which means you can actually separate the ones that have large radius of gyration values versus small radius of gyration values. So by being able to extract these signatures, we are able to say something about the biophysical nature of how these proteins function. But how about putting that together in a package such that the small angle neutron scattering people can use it? So you end up having multiple trajectories that you generate. You do these on harmonic conformation analysis, which leads you to separate these high and low energy states or high and low radius of gyration states. And then what you end up doing is taking this entire data set and putting it down to a very small number of clusters, which tells you how these atomistic ensembles look like. So you take the simulations and project them down to an extremely low dimensional space, which then allows you to recapitulate what happened in the experiment. So on the extreme corner of the right-hand side, you can reconstruct these conformational states with respect to the experiments. And what you cannot see clearly is that I have overlapped what we get from the experiments and put them on top of the MD simulation. So that is the theoretical scattering curves versus the observed scattering curves. And they actually do a very good job of recapitulating what the experiments do. But that is not the key thing there. You can do that with data analysis, which is kind of common in biology. But the thing is, we wanted to say, why not we fulfill the cycle? So basically, how simulations and data analytics can drive the experimental data forward. So one of the things that you can do very nicely with small angle neutron scattering is that you can speculate, or you can basically identify different spots on the protein and deuterate them, which means that you can label them with different experimental data. And by doing that, you get a collection of these experimental data, which then can be fed into our data analysis framework in such a way that you can complete this cycle. So ultimately, this resulted in improving the SANS resolution itself, which was a big deal for the biologists that used to come and work there at the lab, by which they could use the analysis to drive their experiments forward and propose new hypotheses, which could eventually allow them to understand better of these physical phenomena that they were trying to find. And one of the more practical advances of this is that we were able to use the same strategy to look at drug binding. And in this case, this happens to be a molecule which is P27, which is implicated in a large number of cancers. And one of the things that we could do was we could take these simulations, which was about 15 to 20 microseconds long, and design molecular scaffolds on which this binding could be done. But the problem is you have all these simulations. If I don't tell you where the experimental data matched, you won't have any idea of whether it matched or not. So you could reconstruct a very small storyboard that the biologists could play again and again and see what happens with respect to the different parts of the protein and how they're interacting with each other, and put that together so that they could construct better wet lab experiments and guide experiments further. So let me go into the next scale, where we are working with genomic scale data. And in this case, one of the things that we are trying to do is understand how ecological factors, especially climatological and environmental factors, affect virus evolution. And in particular, you have heard all of this in the modern day literature. You have Zika virus, you have Ebola, you have West Nile. And one of the things that we noticed was, if you look at the number of West Nile virus incidents, you can see these anomalies across the country, where you basically see that whenever they have had higher winter temperatures, then you have higher West Nile virus incidents. In a similar way, you also see when you have anomalies in precipitation, if there is more rain during the springtime, you also see a higher number of West Nile virus incidents. So this is actually a picture that basically summarizes that type of data. But if you track how this virus evolved over time, something very important was basically this virus over a very short course changed its type of evolution. And one of the questions that we wanted to ask was, is this changing as a consequence of climatological factors, including change in weather patterns, environmental data, which includes chemicals, particulate matter, and so on. But you cannot stop there to understand how this virus is evolving, but you have to actually keep track of how the virus itself is talking to its neighborhood, which means that it is talking to other bacteria, which is the resident microbiome that you see there. And there are also transient microbiota through which it interacts and has all these different changes. Thankfully, at Oak Ridge, we also have the ability to collect this type of data sets where we have a large collection of environmental and climatological data. We have genomics and panomics data, which has been collected at quite high resolution. But putting this together in order to extract features out can be quite painful. So some of the data sets, like the climate data, is about 600 terabytes or so, which collects over 50 years of simulations and also data collection from the various sensors around the country. Environmental data, which has been collected by EPA, is approximately expected to be around a petabyte. And the genomics and panomics data are also quite large, which is over several terabytes of data. And now you have all these structured and unstructured data sets which you need to put together in order to build approaches. One of the first things that we attacked, though, was to find out what species are present even within a given sample. So within that viral and reservoir sample, we wanted to find out what species are present. We wanted to build techniques that would allow us to extract these metagenomic signatures so that we can understand how different populations are talking to each other. And then we ended up building techniques that would allow us to visualize this information in a meaningful way so that we can understand how not only the microbial structures are present, but we can understand what are the other interactions that the virus is having with respect to the different species in that sample. And then one way to visualize this is to construct a phylogenomic network, which we call it as a dtree.ornl.gov. If you're interested, you can go check that out, where you can visualize the entire evolution of Ebola since its start until the end, till last year. And it'll tell you all the different species that are present with it and so on. So now you're bringing together different techniques that have to reflect on how we analyze these data sets and also visualize them. So this is one of the exciting problems that we are working on in the genomic area. And to give you a brief overview of what we are doing with the big neuron challenge, big neuron happens to be high throughput imaging in order to map out neuron morphology reconstruction. So if you take the billions and billions of neurons that are in the brain, they are not stratified into like more than 30, 40 different types of neurons per se. But the overall architecture through which they're connected inside the brain dictates their function, and so it's very important that we understand this neuron morphology somehow. So this is a community-wide effort that's been going on with multiple labs involved in it, where they're contributing neuronal reconstructions across these data sets of about 30,000 neurons for which there was manual curation done. So a manual curation, to give you an example, even a small neuron, if it is just a small pixel image, it can take anywhere between a couple of hours to a couple of days to process this data. So they have painstakingly done the work on about 30,000 neurons or so, but we want to automate this process. But before automating it, you need to set up standards for saying whether this method works or not. So in order to do that, we have to establish some sort of a protocol that allows us to compare these algorithms and also establish a big data resource that others could come and use in order to do this type of work. So how does big neuron look like? It is essentially something that has been putting together with 4 supercomputers across the country, where we are constructing these neuronal data sets using neuron images from over 30,000 standard forms, and the data will be eventually hosted across the world in 3 different mirror sites. So really speaking, why did we use supercomputers for this is that you have millions and millions of images on which you want to do the processing, but you also want to simultaneously visualize this data somehow. And if you think about this, is actually the raw data that comes in. Each of the algorithms outputs a different reconstruction. Then you construct a consensus reconstruction from all these algorithms and compare it with how the human thinks about it. So this is one of the first times where we could use the Everest visualization system inside ORNL to look at these types of data sets simultaneously. So if you have 26 different reconstructions, we could build all of them together at the same time, fetch the data from our HPC unit, and look at all of them together so that we could see if we were doing the right thing or not. So last part, which is focused more on population health. In this case, we are more interested in understanding, say you have all these electronic medical records, which is actually another data set which is quite large, and you have extremely detailed data of how the patients were treated, the laboratory, the vital diagnostic procedures, and different types of operations that were done on the patient. And we wanted to find out, given these medical records, is it possible to find sequentially ordered data sets on which operations were carried out? So in some sense, finding it out in an unsupervised way, how patients are being treated by their doctors. So heart disease patients have certain protocols that they have to follow. There are certain procedures that need to be done for mental patients and so on. But we wanted to see if we could use this to extract these types of patterns. Of course, we were able to show that the medical records are able to recapitulate what we know from flu outbreaks and so on. But to give you an idea how this data set looks like, so if you have a patient, he goes to his primary care physician or the doctor, and that provider submits a claim on behalf of the patient for this to be processed. And eventually it goes through a clearinghouse, which is where the data is reported. And in a similar way, a pharmacy event is also recorded that way. But in each case, you're recording all this information, which is quite vital: the patient identifier, the date of service, the code of service that was performed, the procedure that was performed, and so on. And it has over 1 billion records every year, which covers about 25% of all the physicians on the nation, and about 2/3 of all the patients in the country. In a similar way, you have the pharmacy data also, which records over 3 billion reports a year. 70% to 80% of all the dispensed pharmacy records are processed, and about 200 million patients again. So if you had this data, the key question was, could we use this data to do public health dynamics? Could we ask questions on saying how diseases are spreading? So one of the first things that we did was to look at the pandemic flu season of 2009-2010, and here is a movie showing to you how many patients reported with flu-like symptoms across the country for the year of 2009-2010. So the date is actually tracking it. And what I want you to pay attention to is what zip codes actually light up and how many patients do show up at each of these places. And this is around October, where you can see it's pretty much covering the entire country. But the problem is, if I just show you this movie, all you can say is, okay, in October I saw a peak and then it ebbed down and nothing else. There's this challenge of extracting meaningful spatial and temporal patterns from this data, and how do you allow this interactive visualization to proceed from the data forward. So in this case, we developed Orbit, which is actually something that is sitting on top of a technology stack that is built entirely on open stack architectures. And that allows us to build these nice visualization tools to analyze these data sets. So Orbit is essentially a platform to do EHCR data analysis for public health surveillance. And we have used these inputs, whatever patterns that you extract, the spatial and temporal patterns, to drive simulations forward to monitor what-if scenarios, so that we can understand whether a particular flu situation needs to be tackled in a particular way or not. So this was actually a tool that was highlighted as part of a DHS, Department of Homeland Security Technology showcase. And finally, we were also able to extract sequential mining from EHRC data in order to find out how patients were treated across the entire country. So in summary, data science is actually central for quantitative biology and biomedicine. So we need to develop new algorithms that involves theory and algorithm side of research that has to be interfaced with scalable tools so that you can engineer them to be successful. And these have to be hybrid in such a way that they can provide seamless transitions between programming and deployment environments. And finally, having visualization tools that can meaningfully allow even novices to interact with the data such that they can look at interpreting how they can use this information to drive hypothesis-driven search. And I've given you different vignettes of how we are using supercomputers and facilities across the experimental labs here to do data-driven simulations and also integrate them with experimental observations. Final thing, a little bit of shameless self-promotion, which is that we have this conference for brain informatics and health, which is being held in Omaha, Nebraska, If you're interested in submitting a workshop or a tutorial, please do submit. This year we are co-located with the Web Intelligence Conference, so there's a whole lot of people that are coming out there. Last year we did this in London, and it has about 1,000 people. So you can ask the rationale between moving from London on one side to Omaha, Nebraska on the other side, but that's where we are. So if you're interested, please do send me an email or a note. We can see how you can participate, and we would definitely be happy to have you there. And with that, I would like to thank all the people that worked with me and also the funding sources, and happy to take your questions.
A great talk on computational biology. Any questions for Arvind? Jack?
I always have one. So Novartis and some of the larger pharmaceutical companies are doing at least parts of the drug discovery in public cloud, particularly looking at analyzing small molecules, millions at a time. You know, out of all of this, what part can cloud computing play in this whole scene?
So cloud computing is a very different beast. I think the requirements in biology for cloud computing are very different, in my opinion, because one of the things that we have found is that unless you're co-located with a biologist and you help them understand the data, they're probably not going to pay much attention to any sort of cloud environment, or they actually don't care whether they're running on the cloud or whether they're running on the HPC platform. They just need an answer. And if that is your goal in order to build better tools that educate them, it doesn't really matter how the deployment aspect happens. It can be on the cloud, it can be on the HPC platform. HPC plays a very important role when it comes to doing these simulations. And if you were to run molecular dynamics simulations on the cloud, you can quickly start running costs just because you're intensely using them. But the computing costs are really not that much, so it doesn't add any benefit to it. But of course, there are some advantages to running things on the HPC platform, because you have the data co-located with the experimental facility, which can utilize the power of both of them. Otherwise, you end up transferring a lot of data back and forth, and that can be quite a bottleneck.
Anyone else? Questions? Susie.
Thanks. You gave examples across different scales. And I was wondering if, just your personal opinion, you think one of them is more ripe for breakthrough than the other? Maybe because more work has been done there, or there's just more excitement?
Personally, I think I would stick with cancer, because that's where most of the work in terms of HPC is kind of mature. The drug discovery algorithms and so on, they're not as mature as we would like them to be. They're much more suited for the cloud type of environments. But when it comes to probing these systems that are kind of very amorphic in terms of the protein sense, that is something that cannot be really done right now on cloud-like environments, because of the nature of how many simulations you need to run and how large these simulations are and how much time they take and what pains you need to take in order to go there. But my preference would be on this side. But from working on the neuroscience data sets also, which we have a lot of interactions with, and being part of the neuroscience community, we have started seeing a lot more excitement around it. But this is on the European side. The excitement in terms of simulating and so on is on the European side, but our sides are more focused on the data analysis and the data tools that we need in order to understand the experimental data that we are generating. So.
Thank you.
Thank you. So we're going to take a quick break.
There's a question.
Oh, there's one more question. I knew Jack was going to ask something.
So on the neuroscience side, there's a lot of challenges in trying to classify the different types of neurons and identifying it from the images that you have. So when you were looking at the images, were you also classifying the types of neurons to try to determine how to sort them out, and how well did you do?
It depends on who you ask. So it's actually a classic question that we get asked often. And one of the things that we are trying to do is, from my side, my personal interest side, I'm not involved in the algorithm development, but I'm involved on the side of managing the data and putting it together so that it can be used. But it depends on who you ask in terms of what the benefits are in terms of the accuracy rates and different types of neurons and things that we are working with. So if you ask the neuroscientist, a certain algorithm performs well on astrocytes. If you ask a certain other person, he says that no, this works on general all types of neurons and it does very well on these types. But there is just too much of uncertainty around it just because of the nature of how these data sets are accumulated. So for example, even in the 30,000 neurons that we have, the human data is probably about 1,000, not more than that. And many of them are from just one part of the brain itself. They're not from all across the brain. So I don't have a good answer for you in that sense. But what we have worked with and what we have seen so far is quite encouraging in the sense we are able to put together a platform through which we can disseminate these different algorithmic tools. And eventually we hope that they'll be able to run it and see whether it works on their data sets that they're generating. And having people to contribute to a central resource for data is actually something that we are constantly interested in, and we are trying to encourage them to do.
All right, great. Well, let's thank Arvind, and we'll be back in about 20 minutes. So there's coffee and snacks at the back. So please come back in the room in about 20 minutes so we can continue the data analytics.